Okay, so we're going to move into the Cercospora part of our talks today, and our next speaker is Dr. Nathan Wyatt. He's going to talk about CLS Epidemiology.
Hi everyone. Glad to see everybody got here safe today. I know it's slick out there. I almost biffed it on my way into the building. I'm going to talk to you guys a little bit about some of the molecular techniques we're deploying to monitor Cercospora leaf spot disease progression, as well as tracking fungicide resistance mutations and seeing how we can use that data to best manage Cercospora leaf spot during the season. So, as a brief introduction of Cercospora leaf spot, it's caused by the fungus Cercospora baticola. It has a polycyclic life cycle, and that's an important factor for managing this disease to keep in mind. It's extremely genetically diverse, which means it can adapt very quickly. So when you're thinking about adaptation to management practices, diversity is adaptation essentially. And it's primarily controlled through fungicide applications and host resistance. And there's a famous picture up here that I believe Mike Metzger took of a field where a grower stopped his application, waited until after the weekend to come back and finish it, but it was already too late. So generally speaking, the problem facing fungicide or facing CLS management is fungicide resistance. And this is the 2023 data from the Red River Valley looking at the percent prevalence of resistance based on a field-by-field basis. And you can see that we have fairly high resistance levels to the majority of these fungicide chemistries, with the exception of tin, which it's nice to see that we had a break in the high levels of tin resistance this year. And then one of the interesting trends we see here as well is that Inspire and Provosol have lower fungicide resistance incidences than Eminent and Proline. And then Headline, as we're very used to seeing, has high levels of resistance across the board. Now, to qualify this is all done at the end of the year for our end of the year surveys, and I'm going to be able to show you how this actually fluctuates throughout the year here in a couple slides. When thinking about managing Cercospora leaf spot, it's a good idea to keep in mind the disease cycle of this pathogen. You can think about it in these various stages here with pre-infection, latent infection, CLS symptoms, and then the polycyclic cycle. And so pre-infection would be everything prior to beets being planted. So the snow melts off the field, soil inoculum is exposed, starts blowing around. Eventually we plant, and then there is leaf material for those spores to land on. Latent infection kicks off, and we have some pretty good results from Dr. Secor's lab and my own weather modeling that shows that we typically detect latent infection starting when we see temperatures and environmental conditions that are conducive for spore germination. So that would be temperatures surpassing 50 degrees Fahrenheit and the availability of free water. And you can think about that as rain events in the late May section of the year, or events that lead to prolonged dew formation on the leaves. So I know lots of you walk through sugar beet fields, and if you ever end up with your pants wet up to your mid-thighs, that's a good CLS conditions. And then you have this latent period from when that infection starts to when symptoms develop. And when symptoms develop, you're starting to get new spore production, and that's really where you start facing an exponentially more difficult problem controlling Cercospora. Because as each lesion that you see will produce thousands of spores, and those spores will blow around, causing new infection, producing new lesions that then produce new spores, and thousands of spores build on each other to make a pretty big issue. And the part that's not on here is the end of the season residue management definitely plays a major factor here. But I'm here to talk about latent infection. And as just a quick reminder of what latent infection looks like, in greenhouse assays when we inoculate sugar beets at 7 days post-inoculation, which is the top panel there, you can see that sugar beet leaf looks pretty healthy, but when we zoom into the cellular level and stain the plant cells blue and the fungal cells green, you can see that there's a bunch of green in there sort of intertwined with all the plant cells. And this gets worse at 10 days post-inoculation, and then eventually we see symptoms at 14 days, given nice humid conditions for the pathogen here. So I use this as an example to show you that even when you're not seeing symptoms, there's a fairly comprehensive infection that's going on, and that's what we're trying to detect with our latent assays. So for the last few years, we've been running this latent infection screening on 280 fields throughout the Red River Valley. 56 of those fields come from southern Minnesota. And we run a molecular qPCR assay to detect mutations for resistance and sensitivity to strobilurins, so your QOIs, DMIs, so your triazoles, and then benzimidazoles, which you can think of as topsin, is what we typically see. And generally speaking, for southern Minnesota, in 2023, this is what prevalence looked like for latent infection. Our first samples came in June with fairly low incidence and steadily rose until the week of the 19th of June. And then we had this huge spike from the week of June 19th to the week of June 26th. And so this is a really nice target zone for us to look at weather conditions that would promote this big jump here, because we know that spores are present. And as far as what this looks like compared to the rest of the Red River Valley, this corresponds very closely to what we see, where so week 5 here, W5 would be that last week of testing for southern Minnesota. We did a few more tests in week 6 for the very northern regions of crystal sugars growing region, which they just typically see a slower progression of latent infection onset. But generally speaking, we see near 100% positivity of samples towards the last week of June, first week of July. And that corresponds fairly closely to row closure in those regions where that's happening. Another way to interpret this is that the pathogen in this case has adapted its infection cycle so that it's infected a majority of the growing space asymptomatically right as we get to row closure, which then it has a nice little microclimate to really be a successful pathogen. Now, because we're tracking fungicide resistance profiles as well with this detection assay, we get some interesting results as far as the differences in fungicide resistance prevalence for the different chemistries. So one of the interesting things we see with strobilurin resistance and sensitivity is that sensitivity shows up first, and then strobilurin resistance comes on a little bit later. And we're fairly interested in why this happens. Some greenhouse experiments we've done, laboratory experiments we've done show that strobilurin sensitive isolates sporulate more. So more spores, they sporulate faster, and they sporulate at lower temperatures than resistant isolates. So this might be why we see this sensitive wave come on first, because temperatures haven't risen enough for the resistant isolates to start kicking out spores, and then we see a lag phase to that. But this could also be influenced by other management practices, everything from rhizoctonia, fungicide applications, to other cropping systems, because you have to remember the field you're planting in is not necessarily the field where the inoculum is going to come from. There's plenty of other fields that were sugar beets in the previous year, and that's where a lot of your inoculum is going to blow over from. For triazoles, focusing on eminent and proline, we see an interesting trend where resistance actually emerges first and sensitivity lags behind. And this, in laboratory experiments, with triazole-resistant isolates, we see the opposite effect that we do with strobilurin-resistant isolates, where triazole-resistant isolates actually seem to grow faster. We haven't looked at sporulation, but these certainly are quicker to grow on laboratory plates. And this might be why we see resistance emerge first, but then this sort of equilibrates with resistance and sensitivity reaching a fairly even frequency and then skews very quickly to resistance again. So this, the way I would interpret this is that you have resistant isolates showing up first, sensitive isolates catch up, but then the resistant isolates still manage to outpace those sensitive isolates in our detection. When we look at the other triazoles we are able to detect resistance for, so Inspire and Provosol, we see a slightly different pattern in that you eventually end up with a fairly even ratio of resistance and sensitivity moving into the year. And one thing I didn't point out here, red is resistant, blue on these graphs are sensitive lines. So if you need me to go back on that later, let me know. For benzimidazoles, we detect high levels of resistance heading into the year. It's important to note, though, that in this, for topsin, we're only running a probe to detect the resistance. We don't run a sensitive probe just because we can't fit all of that into one assay. And so even though we have high levels of tops and resistance, we still don't actually quite know what sensitivity levels look like, because many of these samples, because they're field samples, can be mixed with multiple isolates. It's possible you have more like a 50-50 ratio of resistance and sensitivity, or it could be exactly what you see here and everything seems to be resistant right off the bat. But that's something we're going to actively look into in the next couple years as well. Now, you might be wondering, why do we care so much about latent infection, other than the utility of knowing what the fungicide resistance profiles might look like running into the application period. And what you see on this graph here are some data we've been working on with crystal sugar, where we've binned fields into categories based on the first week of the year when fungicide application started. And so one of the things you can see, and the other thing I should point out here, is that this has all been normalized to the fraction of recoverable sucrose per acre based on the maximum for each of those bins. So the highest number from each year was set to 100%, and then everything else was measured as a fraction of that 100%. And what you see here is that if our highest recoverable sucrose across these acreages occurs if we apply a fungicide in that last week of June, which is just before we detect 100% prevalence of CLS across their region. And then you can see that there's, from the last week of June to the first week of July, there's about a 4% reduction in recoverable sucrose. That's actually fairly stable across July, and then as you head into the last couple weeks of July and August, that starts to drop off again. And so this is telling me that by controlling Cercospora early in the year, we're actually preventing a larger issue for the plant to deal with, and that's resulting in a higher, or an increase in recoverable sucrose. So from an economic perspective, if you take the highest recoverable sucrose in your region, and you're able to kind of calculate how many dollars per acre that means for you, you can go through this chart and balance out where you think you land and whether or not an additional fungicide application early in the year would be economical for you. Or wherever you land in this chart, you can do the sliding adjustment. So now I'm going to kind of pivot and talk about shorter timescales. Because we have this molecular assay and it's been useful for latent infection, we were also interested in looking at how fungicide resistance changes throughout the year, especially following fungicide applications. And a lot of this was spurred by what you see here. So using strobilurin resistance as an example, in the black line from 2012 to 2023, you see this increasing prevalence in strobilurin resistance, and this is why we think headline isn't typically a very useful fungicide. However, at the beginning of the year, we see a much lower prevalence of resistance to strobilurins. And this difference from the beginning of the year to the end of the year got us thinking that maybe this is more dynamic throughout the year. So based on sort of a pilot study and a backup study with American Crystal Sugar on fields that had fungicides applied in this order, and using strobilurin resistance as an example here, in 2022 we saw this pattern of resistance, and then in 2023 we saw this across 8 fields as well, where our strobilurin resistance was approximately at a 50-50 ratio on average. We applied it, when a DMI EBDC fungicide application was applied, the strobilurin resistance dropped quickly, and then it gradually rose as the year went on. Interestingly, the tin tops in application didn't seem to actually affect strobilurin resistance. We're not quite sure why strobilurin resistance trended up after the initial decline. It could be very much like what we see at the beginning of the year. There's other factors involved. Other crops have strobilurins applied to them. And those could be changing the inoculum profile in those fields and then drifting over onto sugar beet fields, but we're not sure. And then what you see at the very end here is when a strobilurin, so headline, goes on that field, all of a sudden you get a big spike in strobilurin resistance. And then we sample for our end of the year testing in September, and that's why we detect high levels of strobilurin resistance. It's the last thing we filter our population for. Now, looking at this across the 2 fields we conducted this project with for southern Minnesota, so southern Minnesota so Mark Bloomquist and David Metler were able to help pick 2 fields to track this across, and they sampled quite a bit throughout the year. And you see, looking at strobularin resistance frequency, that it's quite variable throughout the year, right up again until you apply a strobularin at the end of the year, and then you get a big spike in resistance prevalence and a pretty big drop off in sensitivity, which makes a lot of sense. Because this is a single year of data on just 2 fields, this being only one of the fields I'm showing here, I wouldn't feel comfortable making any conclusions based on this other than it's exactly what we expected. This is highly variable across the year. When we look at the second field, quickly pivoting to trizol resistance across that same field, we see a big spike in trizol resistance for this particular field right after we apply a trizol. So DMI-EBDC combination goes on and you get a big spike in trizol resistance. That tapers off throughout the year to almost a 50-50 ratio again before slowly trending up, and then following the last DMI application on this field, you get a little spike there. For the second field we tracked this on, you see a pretty similar pattern. There's some variability in strobularin resistance in response to the applications that are going on that field. And then, interestingly, at the end of the year, the spike to resistance happens even though a strobularin didn't go on that field. So this is an interesting event, and we're curious why this happened, and we're trying to work on some hypotheses to test why this might happen, whether that's based on inoculum from surrounding fields or new infection that's occurring at that time of the year that's mostly resistant thanks to broadly applying strobilarins. When we look at trizol resistance, we see a similar pattern that we did before. You get a big spike in resistance following the trizol application, and then that tapers off following other applications throughout the year with a slight increase following a DMI EBDC application in between sampling 4 and 5 there. So given what we're seeing as far as fungicide resistance variability throughout the year and end of the year fungicide resistance, we thought it'd be interesting to look at cross resistance. And when we do that with Eminent, Inspire, and Provosol, we see that we have these 2 categories of DMIs. So Eminent and Proline, for Eminent and Proline, we have high levels of cross resistance with about 92% of isolates from 2023 being resistant to both of those. When we pivot to look at the comparison between Eminent and Proline and Inspire, for example, that drops to 60% of cross resistance and 61% when looking at Provosol compared to Eminent and Proline. And then when you look at Inspire and Provosol, so I probably should have had their percent cross-resistant isolates up there as well. So their correlation coefficient is about 0.84, which is a very high correlation coefficient. And when we look at the cross-resistance, it's about 89% of isolates that are cross-resistant between those 2. So really you have these sort of 2 categories of DMIs. And when we look at cross-resistance between different chemistries, so on here this is some testing we did on a select number of isolates for Inspire benlate, which is similar to Topsin, triphenyltin hydroxide, and the active ingredient in the headline. We see low levels of cross-resistance across these chemistries. And the important part about this is that this is a great argument for tank mixing with multiple chemistries, and this is also a great argument for rotating your fungicide applications. When you can think about it this way, if half of your population is resistant to tin and half is resistant to a DMI, in your first application, you're killing half the population. The tin population will blow up, and then you'll follow up with a tin and kill that whole population. So the same can be said for tank mixes. So if you're spraying, a good example would be like tin Topsin. Cross resistance is pretty low, so in a tin Topsin tank mix, you're likely getting the majority of isolates infecting that field if the application is successful. So then there's weather events that factor into whether or not those are successful. And if we could control the weather, we'd really have this under control. So just to quickly cover a couple takeaways from this, CLS latent infection prevalence reaches nearly 100%, and that's in 2021 and 23, it corresponds pretty closely to row closure. So when you start thinking about the economics of when to apply a fungicide, the early July would be a good start date, and some of our data from further up north shows that maybe even that last week of June. Our molecular assays can be used to detect this latent infection as well as fungicide resistance. I'm happy to say that we've got a new machine in that can do more mutations in a multiplex. So this year we should be able to do strobilurins, DMIs, benzimidazoles, and we're testing some tin resistance markers to be able to track that. And we're just waiting on a little bit more validation for that. And then the last thing, or not the last thing, but fungicide resistance is not fixed. So management practices influence the development and maintenance of resistance in this pathogen population throughout the year. So even if you see high levels of resistance developing over time, it's concerning, but we absolutely have ways to adjust that and pivot and manage CLS in the face of resistance and hopefully then modulate that resistance backwards. And the other piece of that, which relates to the previous point, is that cross-resistance remains pretty low between fungicide chemistries. So tank mixing, rotating your chemistries, that's going to be your best tool from a chemical perspective for managing CLS. So a couple big thank yous to Mark and Dave for helping with all that sampling, and then our Fargo team here, and all of the folks who have helped out with funding and support for these projects. So I can take any questions you might have, if I have time.
Absolutely, you have a couple minutes. Any questions for me?
Do you ever use hydrogen peroxide?
So the question was, do we ever use hydrogen peroxide? We use hydrogen peroxide to pre-germinate sugar beet seeds if we're doing assays on seeds, and that seems to kill everything. But I don't know that anybody's ever tested hydrogen peroxide as a fungicide application, as far as I'm concerned with our results. Yeah.
There's been people who have tried it. The problem that we've seen, it does what it does that minute, and then a half an hour later, it's all over. And so anything new that moves in has a fresh start.
One other thing too on that is that fungal pathogens, so a part of plant resistance is typically this release of reactive oxygen species, so hydrogen peroxide would be a good example. So pathogens already have to sort of deal with hydrogen peroxide and reactive oxygen species, so likely that mechanism is built in. It's something that we've looked at as far as an adaptation to host resistance, but we've never, I've never tried it in like the scale you would for fungicide applications, if that makes sense. That's a good question, though. Other questions?
What's the likelihood or time scale for which these isolates would develop cross-resistance?
That's an excellent, so question was, what kind of time scale are we looking at as far as developing cross-resistance? And that really, it's hard to put an exact number on that. It depends on how it's used. So if you're very consistently, let's say you're constantly doing DMI 10, DMI 10, DMI 10 with no tank mixes, it's possible that that would develop fairly quickly as long as you were using the same chemistries, but really we don't know. It's something we're working on with Dr. Secor's collection of isolates from 1997 to present. You know, there's variable levels of resistance to all of those, and that time span incorporates the introduction of headline and eminent. And so we're going back and sequencing those and looking at, okay, how long did it really take for resistance to blow up and become a problem? So hopefully we'll have some answers for you in the next couple of years.
Where do the other triazoles, the enables, top guards, where do they fit in this cross resistance? I mean, we got six different ones in the bucket, and we're testing 4.
Yeah, absolutely. So we did just a couple, where we are currently doing some spot checks on with Dr. Secor's isolates from last year with, and you might have to help me out. Febuconazole is one of them, and I believe that's in Enable. And then the one that always escapes me is the active ingredient in Top Guard. But we have that on hand, and we're testing that as well. In previous years, so Dr. Secor has some good data on like epoxyconazole, propiaconazole, ciproconazole, which I believe is another one. But they don't, so what we'll have to do to compare cross-resistance is go back and link everything to tetraconazole, which is eminent, and that's been the mainstay of their fungicide sensitivity program. But since we've gotten that question from Mark and Dave, we're going to go back and look and see if we can link all of those and put them into the 2 categories, or if there's 3. There might be more than 2 categories.
Any other questions?
Sweet, I'll be hanging around. If you guys have any questions, come find me.
Thank you, Nate.